All right. Well, thank you for this. Thank you for the opportunity and for supporting our research, actually. So I'm going to talk about a project that we have in our lab to bring together two separate areas of biology. One part is the leukemic stem cells, which Trang has recently very well described. And the other part is the signaling that may regulate these leukemic stem cells. So, as you were just told, normal blood is organized as a hierarchical situation where you have stem cell populations that can survive for a person's lifetime. And these produce about 1 trillion cells per day. So they produce a subset of cells that are progenitors or transit amplifying cells that then in turn produce all these other cells. And so you can imagine that this is a tightly controlled, exquisitely balanced process. And then some types of leukemia represent an aberrant view of this process. And so you can have a leukemic stem cell, which is a rare set of cells, and they may not be derived from actual stem cells, they just have stem cell properties. And they can be resistant to therapy, and they produce progenitor cells, which then produce leukemic blasts. So all the leukemic cells in the patient. So these leukemic stem cells are really what's sustaining the disease in the patient, and they are what need to be targeted to eradicate the disease and sustain or have a long-term cure for a patient. And so some of the work we've done in the past is to identify genes that are linked to leukemic stem cells and then find that patients who have AMLs, acute myeloid leukemias, that express these stem cell genes at a high level, they do significantly worse. Really reinforcing the idea that there's a correlation between the activity of the leukemic stem cells in a patient and outcome for those patients. Another aspect of biology that we're bringing together with leukemic stem cell biology is signaling, the exosomal signaling. So this is a relatively new field. Signaling between cells occurs all the time. Cells are constantly being regulated. They're told by their neighbors what to do, to grow, not to grow, to kill themselves, to make space for other cells, or if they're out of control, that kind of thing. And generally, there's two main ways that these signals occur. One cell will emit a molecule that'll float over to another cell, either nearby or a little bit distant, and that cell will catch that molecule and it'll get the signal. Other times the cells are beside each other and they have receptors, and those receptors lock and interact, and that transmits the signal. But there's another way for cells to communicate with each other, and that is they can emit little bubbles of themselves. They can bleb out a little package that includes some of their internal components, specially packaged in there, that can then transfer to another cell, fuse with that other cell, and provide that other cell with those components. And this is a way of signaling that's in some ways very unique because it allows the interior parts of a cell to go from one cell to the other. And these are called exosomes, and they can really regulate a lot of processes between cells. And so our question, or one of our projects now, is to see if leukemic stem cells are being regulated or even being signaled to using these exosomes. Because as I mentioned before, we have these leukemic stem cells, and like all cells, they're being regulated or communicating with the cells around them. Either they're leukemic blast cells, the majority of cells, the leukemic cells that aren't stem cells, or supporting cells in the bone marrow that unfortunately support the survival of the leukemic stem cells, especially in the face of treatment. So these cells signal to the leukemic stem cells and vice versa, and that alters the behavior of these cells and regulates them and allows them to respond to changes in their environment and things along those lines. So the question that we're interested in is whether leukemic cells are sending these exosomes to the leukemic stem cells to regulate and support the survival of these leukemic stem cells. And well, there's some complicated slides here, but the end result is what we found is that the vast majority of exosomes produced in a leukemia are produced by the leukemic blast cells. And other groups have shown that in a patient, the exosomes that are floating through the blood, the vast majority of those come from the leukemia. And so it's this very powerful signaling process that's going on in the body. And so the leukemic blasts are the cells that are producing the vast majority of these per cell, these exosomes compared to, say, a leukemic stem cell. And what we've also found is that of the targets of these exosomes, the leukemic stem cell is by far the preferred target. So the exosomes will travel to the leukemic stem cells, and almost every leukemic stem cell in that population will take up an exosome or multiple exosomes, while the more mature blast cells, which really survive, depending on the kind of leukemia you have, only for a couple of weeks, and they get renewed by producing more from leukemic stem cells. While they're producing a lot of these exosomes, they're not really absorbing a lot. Only about half of them absorb it, exosomes. And of those ones that absorb them, they only absorb them at a much lower rate than the stem cell population. And so what we think is that there's a lot of signaling going on between the leukemic blasts to the stem cells. And why is this important? It's important because if these signals are sustaining the leukemic stem cell, or allow it to survive in the face of therapies or things along those lines, we want to be able to target this interaction. And if we can target this interaction, we'll be able to weaken the leukemic stem cells and improve the ability of standard of care therapy to target them. And so in a very basic experiment, just using a simple drug, heparin, we're able to find a decrease of exosomal signaling. So if we expose these cells to heparin, they can't absorb these exosomes very easily. And so that's pretty exciting to us. And in some additional work that we've done, we've looked at the proteins that are on leukemic stem cells that may be involved in taking up these exosomes. And so we've identified a couple of proteins that are present on leukemic stem cells and that have never been shown to be involved in exosomal signaling, but that regulate and allow these leukemic stem cells to take up the exosomes. And this is also very interesting to us because these are surface proteins. And if you want to target a protein to inhibit it, it's best if it's on the surface of the cell because it's much more accessible. And so that's very exciting to us. And so that's basically where our project is right now. We've identified that this signaling is occurring, and it's occurring preferentially to the leukemic stem cell. And we've identified a couple of novel regulators of this process, and we're now looking at if we can inhibit this process and what's the impact of inhibiting this process. And we're doing this all in primary human tissue, and then the intention is to next go ahead and do xenotransplant experiments where we've placed these leukemias in mice and then can interfere with the growth or not of these leukemias in mice. And there we go. So this is my little lab, and these are some collaborators, and I'd like to thank the Leukemia and Lymphoma Society of Canada. Thank you.